Well, hello everyone. Many thanks for attending my virtual talk at the TCII Expo in 2025. I apologize that I can't be with you today, but I have another engagement. Anyway, what I would like to do is just really talk about some research that I instigated in the UK a few years ago, and I have spoke about this once or twice before, but I do have some new information to add, and it's really about this concept, this role of endophytes. And I also appreciate many of you may or may not have heard of an endophyte, and really to me this is just perhaps a whole new way of potentially looking how we can manage some various pests and diseases. So just to kind of set the scene a little bit. We all know, for example, that at the moment in the US, there are some major outbreaks of diseases and pests. And these are ones, this is a slide from a colleague of mine, Dr. Matthew Borden. And what I'm trying to emphasize here is all those pests and diseases on the left-hand side. They're what we call ecosystem changing pests and diseases. Unmanaged, uncontrolled. The effects are going to be absolutely devastating. And it's a similar situation in the United Kingdom, where literally over the past 10-15 years we've seen some really major outbreaks. And the picture there of the cedars going pink is caused by it's a canker disease, a Serococcus. And again, there's no management strategies for this in the UK. We have issues. This is Massaria canker on our London plane trees, where we get this stripe of decay on the top of the branch, and then we have major issues where the branches snap out. And if you've ever been to the UK, you will know that we literally have if not millions of these trees planted within London. So this is a major health and safety hazard for us. This is Pseudomonas bleeding canker, which I'll be talking about later. We find it on our white flowering horse chestnuts, and the white flowering horse chestnut came to the UK 400 years ago. It's done perfectly well, except the past 15 years now. It's absolutely crashing and burning due to this bacterium. This is Cholera ash dieback. And again, where we've had outbreaks in other countries like Denmark and Austria and Germany, you can lose up to 98-99% of the ash trees. And again, it's devastating in the UK. And last but not least, what I'm going to focus a lot of the talk on is this disease. And this is called acute oak decline. And it's caused by a bacterial complex. And we haven't quite isolated all the organisms involved, but we've isolated 2 or 3. And what we see here is these longitudinal splits in the trunk of mature oak. And again, this disease can kill an oak within anything up to 2 to 5 years. But the major problem is, if you've ever visited the UK, is that unlike the US, where you guys have a kind of arsenal of plant protection products, you have insecticides, you have fungicides, you have bactericides. In the UK, we don't. You cannot use any insecticides, fungicides in urban landscapes unless you're a homeowner, and then you can buy it from like the garden centres, the supermarkets. But from a commercial standpoint, there's nothing available for us, and that's because the UK is a very densely populated country, and obviously the potential for these insecticides, fungicides to come into contact with human beings is far greater. So we really have to do a little bit of lateral thinking. And just to show you the issue, this is an insect pest that came into the UK about 20 years ago, horse chestnut leaf miner. And this one species of tree composes like 12% of all our trees in urban landscapes. And this is what it looks like come round about July time. The problem is that it has no biocontrol. And again, as a result of this single species of tree is pretty much, it's really had its day in the UK. So how do we deal with these problems? And again, from the UK perspective, what we tend to rely very heavily on is it's sanitation. So I kind of coined the term slash and burn. We literally chop the trees down and set them on fire. And there's nothing wrong with sanitation to a degree. I just feel maybe we have a massive over reliance upon it. And just to give you an example, we had an outbreak of Phytophthora ramorum, and it really attacked the larch trees more than the oak trees. But really, in an attempt to try and stop the spread, I mean, 3 million larch trees were felled, and that was just in the county of Taunton alone, and it really had a very limited effect on spread. So maybe, yes, it should form part of a strategy, but we need to be looking at other kind of ways and means of managing these problems. So again, I just wanted to emphasize these are some of my clients when I worked in the UK. This is Windsor Castle. This is where the Queen, when she was alive, used to spend her weekends. This is Kew Gardens, and again, in the past, I was involved in discussions with some of the management of trees, and we used to do some small scale research projects there. Again, this is in the north of England. This is Capesthorne Hall, and again, a very, very prestigious place. And last but not least, this is actually the back of Buckingham Palace, and the company has the royal warrant, and we're obviously very proud of that. But the dilemma is, when you're dealing with trees within these locations, and here's an example of a horse chestnut tree, one of the Aesculus, and we can see we do have issues. The canopy is starting to go into decline. And then you look at the base of the tree and you think, oh, this was planted in 1917 by His Royal Highness Prince George. And it's kind of quite scary because you're a little bit, man, I really don't want these trees dying on my watch. And then you're treating another tree and you look at it and you're like, oh, good grief, this was planted to celebrate Queen Elizabeth's 80th birthday. It's one of the Ulmus. But again, like I always say, I now live and work in the US, so so really not my problem. But just to be serious, I mean, these are like really rare historic trees. They're in very prestigious places. And I can't just say, hey, there's nothing we can do. Let's just chop the trees down and set them on fire. It's not viable. But as they always say, necessity is the mother of invention. So again, we have to look at different ways and means of managing these problems. So if using any plant protection products such as insecticides, fungicides is a non-starter, what about biologicals? And again, I mean, we know, I mean, normally the words fungi and bacteria are synonymous with disease, but we know microorganisms have been waging war against each other since literally the dawn of kind of time itself, ever since trees came on. These pests and diseases, it's just constant. And we know now there's this concept of using biologicals or plant friendly microorganisms to fight fire with fire. And normally when we think of biological control, we normally think of the option of basically bugs eating bugs. And I'd like to thank my colleague, Dr. Jeremy Sloan. And on the left here, we see a female lady beetle and its devouring scale. And on the right, we've got the lacewing and it's all devouring scale insects. And the thing is, we do have these biocontrol options. But again, and there are situations where it does work, but the situations where it doesn't work, it can be very variable. Biocontrol does have pros and cons. we know that biologicals are composed primarily of living organisms, and a fundamental problem relates to kind of keeping these organisms in a live state after packaging. And I'm not being facetious or anything, but many of these biologicals, to include like the mycorrhizal spores, these types of bacteria that we've started to apply, they're in a sealed plastic bag, and living organisms, they don't live long in sealed plastic bags. They respire, they breathe, so they do have a relative short life. The effectiveness of biologicals is heavily controlled or heavily influenced by weather conditions and soil temperature. And really, we forget these biologicals. We try to attempt or modify soil, leaf, and whole plant micro ecosystems, and it's really complex. I mean, one change in variable, we move into a heat wave, for example, and that can have a massive impact on any biologicals we apply. We move into a drought, that can influence the efficacy. And this is really why the results of biological treatments are often variable, with effectiveness less than is desired. So I'm not ruling them out as an option, but I just feel at times they can't be that heavily relied on. So what I want to do is talk about a very different approach, and I'm going to talk about the research from a PhD student of mine, Oliver Booth. And Ollie is very lateral thinking, critical thinking guy, and he was also involved in the management of a huge oak woodland in Epping Forest in the southeast of England. And we had a huge outbreak of acute oak decline. And as I said earlier on, acute oak decline is, in essence, it's a bacterial complex, and we've managed to isolate some of the bacteria involved, but perhaps not all of them. And what we see over time is these longitudinal splits, these bleeds, which just increase with severity and frequency. But what is interesting is Ollie and I were in the woodlands looking at these trees, and what I want to emphasize is when you look at a tree and you can see the symptoms there with all these splits and these bleeds, but then at the same time, you have trees which are only like 2-3 meters away, showing absolutely no symptoms whatsoever. And again, we see the symptoms here. But if you looked at the tree of the left of that red arrow, you see no symptoms whatsoever. And we thought, this is really interesting. Why is it we've got trees that are so close to each other, and some are absolutely devastated with this disease, and some are showing no symptoms whatsoever? And when we ask this question a lot, is why do we see this? And the standard answer I always get, and when I was in the UK, I was based at a university, and I asked my fellow academics, and they always came up with the same answer. Well, it's genetics, innit? Because that's how we speak in the UK. It's genetics, innit? But I really wasn't that convinced, because we kind of thought, well, maybe there's a different idea. Because if it was genetics, then when you look at those trees, they were all pretty uniform. They were all the same height. They were all roughly the same girth. They were the same canopy coverage. There wasn't any major visible differences to them in kind of size, shape, morphology, architecture, silhouette. And we thought, but if the genetic base is that wide in terms of disease resilience, as in, yes, the tree is absolutely devastated with disease, to a no, surely we would see that genetic variation reflected a little bit more in the size, shape, etc. of kind of the forest ecosystem. But we didn't. So this was our different idea. And if you've never heard of an endophyte, it's really important to emphasize, trees are not sterile. We always, when we cut a tree down and we look down and we look at all the annual rings, we forget that within the tree, there are lots of fungi and bacteria. But these fungi and bacteria, they don't cause disease. They don't do the tree any harm whatsoever. So we call them endophytes. But the key point, inside a tree, it's not sterile. It's literally a living ecosystem. And an endophyte is derived from the Greek words endon, meaning within, and phyton, meaning plant. And again, I'm going to talk a little bit more about this later on and show some actual examples, but there's some real strong evidence that what actually lives within the tree can really be the difference between resilience and susceptibility. Now, a paper came out a few months ago, and if there was one take-home message my talk is giving, please read this paper. You can download it for free from Google Scholar. It is absolutely probably one of the best papers I've ever read from a scientific perspective. And what we're emphasizing is we know trees are foundational components of the terrestrial biosphere, and these play a critical role in shaping ecosystem structure, stability, and biodiversity. We're always talking about the urban forest and the ecosystem the urban forest is, and biodiversity within the urban forest. And again, Dr. Mike Raup from the University of Maryland has done some phenomenal research looking at landscape plantings and how it influences biodiversity. A colleague of mine, Dr. Chris Riley, he's doing the same. He's doing some great research. And again, when we talk about the ecology of trees, we always talk about what lives on the tree. Like in the UK, it's we should plant oaks because you can get over 400 different types of insects. So we plant trees and we look at the algae and the moss and what insects are on them and what birds it brings in, etc. But I really want to focus on what's inside a tree, not what's externally. We've all done a lot of research there. We're all aware of the benefits. And we call it this microbiome research. And this is huge in the medical industry, okay, human kind of medicine. We know that, the microbiome, there's a whole consortia of bacterium and fungi and archaea, and these can have a huge impact on the well-being of their host. And as I say, in human biology, there's been extensive investigations really unravel the implications of microbial biodiversity for like human wellness and disease. And we talk about how, we ourselves, we're full of like foreign bacteria and fungi, and they help us digest our food, etc. And it's really important. But when it comes to trees, and even though there's a lot of microbiome research being done in various environments, particularly grasses, when it comes to trees, which is the Earth's largest biomass reservoir, I mean, we've hardly scratched the surface. We just don't really know what lives within trees. And this oversight really neglects a critical aspect of global biodiversity. And literally, these endophytes are massive key players in tree health and forest ecosystems function. Now, I'm going to ask you a question. And I want you to seriously think about this next question. If I was to ask you, how many living organisms do you think there are within a large tree? What would you say? What would you think that number would be in a large tree? And this information I'm putting up is taken directly from this research paper, which was published in Nature. Okay, so you've got a number in mind. We demonstrate that a single tree can host approximately 1 trillion microbes in its above ground internal tissue. That does not take into account the root system. 1 trillion. That's 1,000 billion. There's 8 billion people on the planet, yet one tree has 1,000 billion living organisms within it. That is absolutely mind-blowing. And you want to know something? I regularly log wood up and burn it and cook burgers and things like that. And it's really affected the way I look at trees, because I know every time I put a log on the fire, I'm probably burning billions of living organisms. And I know it's really weird, but wow. Absolutely wow. And really what these researchers are saying, that basically they use the word plant in the paper. I'd have used the word tree, but they're saying that in essence each tree should be a holiobiont, which basically each tree is a single ecological unit in its entire entity, in its entire self. It's absolutely mind-blowing. And they again estimate that there is one bacterial cell for every 20 plant cells within the wood. But wow, please read this paper. So I want to focus on the endophytes. So just to refresh you, this is what we did. We have trees growing side by side, which again, where we can see symptoms and where we have no symptoms. So what we did is we came along and we took cores of wood from these trees which were showing symptoms of acute oak decline and trees which were showing no symptoms. And we call that asymptomatic. And the reason why it means, even though they weren't showing any symptoms, then maybe they still had the bacteria in there. We just didn't see the symptoms, that's all. But in essence, what we're saying is we took cores from trees visibly showing the disease and cores from trees that weren't showing any disease. We sterilized them and then we grew them on lots of different media. And as you can see, I mean, the amount of endophytes you can pull out of a tree is it was just thousands. Now, I'm not a molecular biologist. I'm not, but my colleagues are. And what we did is once we'd isolated these fungi and plated them out, we in essence we just identified what the species of fungi were that were growing within the symptomatic and asymptomatic trees. We used the PCR, the polyacrylate chain reaction. Again, I'm not really too sure what the ITS1 and ITS4 means. Like I say, I'm not a molecular biologist. Ollie is, but it's just to say this is sort of what we did. We wanted to know was really, is there a difference between the trees showing symptoms of disease and the trees not showing symptoms of disease in terms of their endophytic profile? And this is why I love doing research. I mean, sometimes you get a result and you're just like, whoa, I was not expecting that. So if I was to say to you guys, hey, what type of bacteria and fungi do you think grow within trees? You'd probably maybe say, oh, trichoderma, I don't know, aspergillus, this, that, and the other. And that's the question I got. So again, and at the same time, I'd like to emphasize is, you know, when you try to pull DNA out of a tree, it's not easy. It's not easy. This is maybe why most researchers work with grasses and crop plants. Trees are tough. Anyhow, these are the fungi we pulled from asymptomatic trees. Now, who'd have thought penicillin would be within oak trees? And I mean, not just one type of penicillin. I think we identified 6-7 different species of penicillin within the oak trees that were showing no symptoms of disease. There were even some species of penicillin we couldn't identify at the molecular level. And we were just like, whoa. And of course, Alexander Fleming, production of antibiotics, penicillin. So we're like, wow. So what we did was, so that's the asymptomatic trees. And we did find penicillin in the symptomatic trees as well, but certainly nowhere near the same quantity and nowhere near the same diversity. So we're not saying the reason why some trees are infected and some trees aren't is due to the presence of penicillin. We're just saying, hey, this is one of the factors we found, and it may be significantly contributing. But we wanted to prove that. We had to kind of show that. So, of course, we literally took these penicillin and we put them out on these bacterial mats of at least 2 of the bacteria we knew were responsible for acute oak decline, the Brinaria goodwinii and the Gibsella quercinaceans. But in essence, where it says this one, that type of penicillin is quite happily growing on this bacterial mat. There is no inhibition whatsoever. So it's saying, hey, this penicillin, at least in vitro, in agar, has biological control properties against the bacteria responsible for acute oak decline. So obviously the next stage is to look at inoculation within a controlled environment room. So apologies for the quality of the photograph, but we used bigger trees and we inoculated with the bacterium that causes acute oak decline. Only we didn't use an injection system. We use an infusion system. Believe it or not, it's illegal to inject trees in the UK for like plant protection purposes, but you are allowed to infuse things. So we infused a cocktail of the bacterium responsible for acute oak decline and, again, the endophytes. And these are the bleed symptoms on the inoculated trees with acute oak decline. So surprise, surprise, you know, what you find over time, those cankers develop, those bleeds develop. These are the bleed symptoms on inoculated trees with acute oak decline and the endophytes. And we literally, we came close to 100% control. I mean, you can see the difference. I'll just flip back if it'll let me. No, it won't. Again, just a huge, huge difference. So really what we concluded, because sadly Ollie's PhD came to an end, that they are endophytes within the tree that have huge impacts on the severity of acute oak decline, with the role for penicillins flagged up. We didn't have time to look at into account the bacterial endophytes and also those non-culturable fungal endophytes. So this is the research of another PhD student of mine, Luke Haley. And we took some of these endophytic bacteria and we put them in the presence of Armillaria, a honey fungus. Honey fungus is a major problem in the UK. So we took these endophytic bacteria, the Pseudomonas, and we had 3 antagonistic strains identified, and we identified some Bacillus endophytic bacteria. And again, we just incubated a very simple kind of lab technique. And what we found is in the presence of Pseudomonas, you know, we did reduce growth of honey fungus by about 50%, where with the Bacillus it was about 40%. And Luke was a little bit like despondent, said, oh man, I was really hoping we'd get better results. And I said, hey, well, let's just think about it. Within the tree, you know, these bacteria, it's not like the Pseudomonas says, hey, we're going to live here. And the Bacillus says, oh, that's okay, we'll live there then. You find them in combination. So I said, well, why don't we just mix them up? You know, let's look what happens when you get a combination of Pseudomonas and Bacillus. Wow, when we did, we got up to 85% inhibition. So really what we're saying is that these trees which were having no symptoms of acute oak decline also contain bacteria we know has potential biological control properties against other diseases, in this case, Armillaria. So that is just a little snippet of Luke's research. And what I'd like to do is just talk a little bit about some examples from the literature, because now this kind of endophyte research is really starting to move a little bit, gain traction. Now, this is a disease I'm not that familiar with, but I suspect many of you are. It's basically oak wilt. It's called ceratocystis fagacerum, and it's very similar to Dutch elm disease. It's a wilt disease. It blocks the tree's water conducting systems. Infected oaks die within a relatively short period, within 2 to 4 months. This is a significant concern for forest health. So this is taken from research. It was published by Brooks in '94, and it came from Texas A&M University. And what they did is they isolated endophytic bacterium from several surviving live oaks where oak wilt was endemic. And under laboratory conditions, 183 of these bacterial endophytes isolated showed inhibition of oak wilt. So the 6 most powerful endophytes were then further evaluated using containerized Spanish and live oaks. And what they found was trees that were pre-inoculated with the bacterial endophytes, Pseudomonas dentrificans, again, there was a 50% reduction in the number of diseased trees and 17% decrease in crown loss. So really, again, it's just emphasizing, hey guys, there's a potential here, something we've hardly investigated to kind of look at these endophytes for oak wilt. Again, I'm just taking snippets. 3 canker diseases were shown to be absolute. Their infection rate and severity of canker was reduced when they were in the presence of the endophytic bacterium Bacillus pumilus. And again, that was published by Wren in 2013. So again, we're seeing it against malaria, we're seeing it against bacterial diseases, we're seeing it against wilt diseases, we're seeing it against canker diseases. Obviously, Phytophthora, again, a good paper, 2013. Novel bacterial endophytes from basically the rubber tree as biological control agents against Phytophthora. And again, great results there. These Phytophthora diseases were suppressed using endophytic bacterium isolated from those healthy rubber trees. Again, Ganley 2008, this time against white pine blister rust in Pinus monticola. Again, inoculation of white pine seedlings with native fungal endophytes reduced disease severity caused by the causal agent of white pine blister rust. So again, we know there's potential, and maybe I'm kind of labouring the point a bit. I thought I'd mention again the cocoa plants, because again, we all love chocolate. And again, this is a crop of, it has a key economic return, again, used extensively, has huge issues with Phytophthora. But what they did find was that some of these natural endophytes can significantly, and that's the key word here, these are significant reductions in foliar damage caused by Phytophthora. So again, it's kind of nice to see this research gaining traction. So that's kind of where we are. The thing to emphasize about the penicillin work, we applied it kind of at the same time as acute oak disease. But what we normally find is when we're actually, as arborists, when we're brought in to look at trees, we're normally brought in when the trees are actually showing visible symptoms. So the next stage of the research is, can we use endophytes therapeutically rather than preventatively? And even though I've been talking about fungal endophytes, and I've been talking about the bacterial endophytes, the fungal with Ollie's work, the bacterial with Luke's work, the only one missing is obviously viral endophytes. So again, we're just starting to understand the role of endophytes. We are learning that they also influence stress tolerance and plant growth and secondary metabolites and induced resistance and insect and nematodes. But as I say, what about viral endophytes? So this is research from another PhD student of mine, Sarah Bradford. And I've talked about this Pseudomonas bleeding canker of European horse chestnut. It first found in 2002. 80% of all trees in the UK surveyed have this disease. It's caused by Pseudomonas syringae pathotype Asculii. And what we wanted to know is, why do some trees recover from infection? Why some die? And again, the photograph just shows the amount of devastation. And that's just a photograph of Sarah, whose research I'm going to be talking a little bit about. So what are bacteriophages? Basically, they're virus. Apparently, see, I'm old school. I call them virus, and I get criticized a lot. They're bacteriophages or bacteriophages. I don't know. They're virus. A bacteriophage is a virus that attacks bacteria. And these phages are specific to bacteria, and they break open bacterial cells. So we have an image of a bacteriophage, and that's what they look like under the electron microscope. And really the kind of the groundbreaking research was actually done in the US, where they developed isolation techniques to basically isolate the phages specific to the Pseudomonas syringae Asculii. So this was great. So it meant we could really hit the ground running. We had the tried and tested methodology. And we really wanted to know, is the trees that recover from infection, do they contain these phages? Or do they have higher populations? And maybe it would answer why some trees died and some trees survived. We had 3 sampling sites. We had Reading, where I used to be based, Reading University, Kew Gardens and Harpston Wood. We took these, we tried to isolate these bacteriophages from leaves, from bark, from wood, and from soil. And this is what we found. And again, I'm kind of cutting a long story short. You know, we looked at many types of endophytes, viral endophytes, and 2 really leapt out. And this is, again, how we normally start off in science. What we have here is a bacterial mat. Basically, we just simply plate out the bacteria on agar, and you get this bacterial mat. And then we apply the different types of viral endophytes. And where we start to see these clear spaces, it means the bacterial cells have been lysed or burst open. And we found there was big plaques and small plaques. And the small plaques were caused by a mycovirdi, and the large plaques by a podovirdi. So it was a case of, hey, we've got 2 potential viral endophytes here. So obviously the next stage is to incubate them in more of a bacterial broth, which is ideal for the bacteria to replicate, to duplicate itself. But obviously, if they're in the presence of the endophytic virus, they're not going to replicate as much. And again, we start to see this nicely. The bacterial populations, where there's no phage, you see that orange line. The bacteria, just growth is exponential. But where we put them with a phage, it really suppresses the growth of the bacteria. And again, this is like in agar. So once you've done that, the next stage is then to start kind of using more the seedling experiments, using leaf experiments. And this is the reason why. And it's like, even though the podovirus was giving us great results in agar, in broth, when we actually inoculated on leaf tissue, you can actually see the podovirus, the one on the right. Those lesions, very little difference between the control. And this is a major problem with kind of biological control. Works great in the lab, not so good in real life. And again, this podovirus is a classic example. Again, so not really seeing any retardation of necrosis on the leaf following inoculation with acute oat decline and the podovirus. But with the mycovirus, yeah, great. We're getting great suppression. So straight away we can narrow it down to just one virus, and that's the one on the left where you see the different strains: M1, M2, M3, M4, M5, all causing very little lesion development on the leaves. So we then go to the next stage, starting to use bigger trees where we look at phage, Pseudomonas, bleeding canker interactions. We put little droplets just under the bark. Again, we start to use bigger trees as well. And again, so this is kind of when we just inoculate with water only. So again, because when you inoculate like that, you're going to form a callus reaction. So again, we wanted to take that into account. So that's what happens when we just add water. This is our control where we have acute oak decline on its own. And you can see all the bleeding, again, the lesion development, that tissue breaking down, the blackening of the tissue. And then we have it when we add the phage. Wow, way better. Again, a huge difference. We know those phages are really starting to kind of suppress, manage, possibly even control the bacterium, the Pseudomonas. So again, now we move up into the larger trees. Again, we had 10-year-old, 5-meter tall trees. We had 8 biological replications. When we say 4 sites, that's 4 inoculation sites: north, south, east, and west. But this time we're applying it therapeutically, where we use the Pseudomonas bleeding canker, we inoculate, and then we leave it for 7 days to take hold. And this is more a reflection of what happens in real life when we're brought in and we're like, oh man, the tree's covered in the disease. I'm now firefighting. It's now therapeutic, curative treatments. So again, we left it 7 days so the bacterium could build up, and then we inoculated with the phage. So previous research, kind of more preventative, get it on before the disease. This is actually when the disease has hold. And again, great results. The one on the left is the control, just with the Pseudomonas bleeding canker on its own. Huge lesions starting to circle around the stem. But when we inoculated 7 days later with the phage, it didn't stop the bleeding canker, but it really slowed down the ingress. And you can see a nice big difference. And again, when we quantify that, you know, what we found in terms of lesion size, it was a significant reduction. We reduced lesion size by about 60%. And then also when we looked at the number of bacterium within those sites, we can see the phage treatment. I mean, again, we're looking at 40, so more like 50, 60% less Pseudomonas bleeding canker actually in those wounds following the phage treatment. And I mean, I know I'm going back in time, but just so people just seem to be aware of this research. And this is what, again, Sarah James, she got married, hence changing name. And again, if anybody wants a copy of this PhD, it's in the public domain. It's all out there. Oliver's PhD. It's all out there. So I just kind of wanted to emphasize, I mean, when we think about it, the implications could be huge in terms of pest and disease management. That's all. I mean, what does this all mean? I mean, to me, it means that possibly we can identify resilient, possibly even resistant trees within populations based on endophytic rather than genetic profiling. I mean, we as scientists have really gone down this molecular biology in a big way. And I'm certainly not knocking it. I'm just saying, well, whoa, wait a minute. What about the endophytes? Don't forget, every single large tree has on average 1 trillion living microbes in there. You know, maybe once we've identified these trees, we just use them in breeding programs. That's all. At the moment, we do all these tree crossing, these breeding programs. They're all based on genetics. I mean, what about endophytes to protect against other tree pathogens of concern? I mean, you may remember my first slide, or one of my first slides, you know, with those ecosystem changing pests and diseases. Why not? And And this might sound a little bit strange, but why don't we enhance this, the resilience of susceptible tree species? Why don't we have, take some seed material? Why not get these endophytic populations in solution and then just soak seeds in it, or inject them into young seedlings? And this is not as far-fetched as you would think. This is actually on the go. This is Phaliocephalia scopiformis. It's a foliar fungal endophyte of white spruce, and it produces an anti-insect compound, eugosalin. And seedlings inoculated with this endophyte have increased tolerance to the spruce budworm, Christiunaria, sorry about this, fumiferana. I'm not very good at the Latin names. But this is the key thing. White spruce trees inoculated as a seedling in 2001. 11 years later, after inoculation, the actual endophyte and eugosalin were detectable 11 years later. So it is feasible to inoculate seedlings that will protect them for at least 10, 11 years. And out of 109 samples tested, 100% of the eugosalin positive samples also still had these endophytes. Because don't forget, there's no genetic manipulation here. We are simply taking naturally existing microorganisms that basically medical research has been doing for years and just moving them around a little bit. And what they found was that the mean needle eugosalin concentrations were greater than 2 times that required to reduce the growth of spruce budworm. And again, it's all been published. So it is, there is credibility behind this research. Okay, I want to finish off. And again, what I want to talk about, well, this is what I would love to see as future research. In the UK, we have what we call veteran trees. These trees have existed, they're between 500 and 1,000 years old, and particularly in the oak tree family. And the question is, how have these survived so long? How have they survived to be 1,000 years old? How have they survived acute oak decline? How have they survived sudden oak death? How have they survived armillaria? I mean, we have different Phytophthora and oak trees, Criticola, Cactorum. I mean, is it luck? Is it genetics? Or is it the endophytic profile? I would absolutely love just to pull core samples from these trees and look at the endophytes to see whether or not there's an absolute universal kind of endophytic profile that could protect oak trees for thousands of years. I don't know, maybe it's justifiable. No one's ever done it. So other than that, thank you very much for attending this virtual talk. I genuinely hope you found it of interest. Hopefully you've learned something new. And again, maybe like me, maybe after this talk, you're never going to be able to look at a tree the same again, just realizing that, as you say, literally every single living tree has the potential to contain, I still can't get over it, 1 trillion living microbes in there. So again, thank you very much. If you do have any questions, obviously I'm not at the conference. I'm easily enough to trace down on the internet. I work at the Bartlett Tree Research Laboratory. I'm based in Charlotte, North Carolina. Please send me an email and I'll get back to you. So again, thank you very much. And again, apologies that I couldn't be with you today.